So we will start with you from the topic excitable structures. And before talking about excitable structures, I should remind you about cell membrane. Of course, you learn so much about cell membrane. What is important topic for us right now? Cell membrane. This is double layer of phospholipid. This is important because we can say that water-soluble substances cannot fluent move through the cell membrane. And this is important moment. Because when we are talking about cell membrane structure, we say about transport of different substance through the cell membrane, And general, we can say with use it. All transport through the cell membrane, we can divide for two types. What type? Tell me, please. Who will answer? Type of transport, please. Okay. Type of transport we have with you passive transport and active transport. What is the difference between it? When we are talking about passive transport, we are talking about movements of some substances according gradient of concentration, according to gradient. If we are talking about active transport, we are talking about movement against a concentration gradient with using of energy. So our main idea, passive transport without using of energy, and the same side according to the gradient concentration, active transport opposite. It means that, let's look my picture. What could we see here? You could see here first simple diffusion. Simple diffusion, we are talking about simple substances like example oxygen or carbon dioxide or some easy molecules. They can move through the bilayer phospholipids, and they move from the higher concentration to the low concentration. Second, facilitated diffusion. Facilitated diffusion through the non-specific transporters and through the specific transporters. What does it mean? It means that other substance, how I told you before, they are not able transported through the cell membrane without special helpers. These helpers, this is protein channels. Protein channels, you could see here, look please. You could see here, we have with you protein channel. Through this, or specific or non-specific, through them can move some substances. But again, we are talking about passive transport. Cause of passive transport, we are talking there moves from the high concentration to the low concentration. And from the right side, you could see active transport. Active transport, we are talking about movements against the gradient of concentration. For example, you could see, for example, sodium. I will draw for you. Sodium, high concentration outside of the cell, low concentration inside. And sodium can move to outside only cause of active transport. Special transporters, special pumps. Pumps always works according with using of ATP energy. This is first step. Okay. Let's move. Ion channels, what could we have inside our cell membrane? Different types. Let's look. Ligand-gated channels. Ligand-gated channels, it means for the opening of this channel, we should have connection with some ligand. For example, neurotransmitter. For example, I don't know, what kind of neurotransmitter you remember? Да.
Siempre.
For example, acetylcholine, epinephrine, norepinephrine, serotonin, dopamine, these all neurotransmitters. They, for opening some channels, they should contact it to the special receptors, and after we have is the opening of channels. Second type, mechanically gated channel. This type of channels we have been studied with you when we were talking about, for example, sensory system, about somatosensory system. When we touching to our skin, we feeling with you this touch. How is it working? Because of mechanically damaging or irritation of our mechanoreceptors, under pressure, channels are open and ions moves inside. Always open channels, they're working for excitable structures like muscles, for example, or neural cells. And in state of rest, when we not touch our cell, when the cell doesn't have any irritation, these channels are open always. And voltage-gated channels, this case we're talking about some electrical irritation or some electrical changes outside cell membrane. Possibility of the cell to be irritated, named as excitability. Excitation, this process on the membrane of excited cell during irritation and accompanied by changing in membrane potential. Irritants could be external and also internal. External, physical, chemical, biological irritants. Internals, it's of course about hormones or neurotransmitters, mediators. Excitable structure, I remind you, excitable structure, these three types of tissue. It's nervous tissue, muscular tissue, and secretory tissue. This structure, this tissue, we named as excitable. Why they are excitable? Because outside of their cell membrane and inside cell membrane, we have difference between potential. If you will try to find inside your memories, you will find next things. In the state of rest, outside a cell membrane of excitable structure, charged as positive. But inside, it's charged as how? Guys, if outside positive, inside negative, right? Inside will be negative. We are talking about condition, state in rest condition. And we say that we measure membrane rest potential. I remind you, this is potential difference between external and internal surface of cell membrane in state of rest. when we not have any irritation of the cell. MRP, membrane rest potential, remind you, in excitable cells is from -60 to -90 millivolt. What does it mean, this minus? It means that any measurement of MRP or action potential we created inside of the cell. And we say that in state of rest, here is -60 or till -90. It depends on different tissue. Depends on different tissue. Also, we should say mechanism creation with MRP. How does it happen that outside and inside of some of our cell we have this difference in potential? It happened because of few factors. Few factors, first of all, we should say about difference concentration of ions. Three main ions we have with you: sodium, potassium, and chlorine. Sodium, potassium, and chlorine. For a theoretical calculation of membrane rest potential, we can use Nernst equilibrium potential formula. This formula calculate and using next numbers: -60 and logarithm concentration, some ion, inside of cell and outside of cell, internal and external. But when we are talking about creation of MRP, we are talking about three main factors. Three main factors. First, ion concentration gradient. Look, we say that, again, I remind you, that outside of our cell, we have high concentration. Which ion? Outside cell, which ion high concentration we have? Say again. Sodium. Yes, sodium. You see, sodium is outside high concentration. Sodium. Inside, we have high concentration potassium. Potassium. I pay your attention that on sodium and potassium, we have positive charged ions. Both of them are positive charged. Also, outside, we have higher concentration of chlorine. But in the state of rest, I remind you that outside our cell membrane charged as plus, but inside is charged negative. Negative. So the main question: who create this negativity inside of the cell? Who created? What is the mechanism creation? We say with you that, first of all, this different concentration, okay. Second factor, this is permeability of cell membrane. permeability of the cell membrane. For you will easier understand this process, please look this scheme. Outside sodium, you see, it's like circle. Inside potassium, it's like triangle. Permeability, it's physical characteristic. It's mean how many channels, how many holes we have in the membrane for one or other one ion. And what could we see here? For which ion higher permeability? According to my picture. Guys, for which ion higher permeability? Look, triangles and circles. Higher permeability you could see for potassium, not for sodium, but for potassium. One second, please. Is it better you can hear me now or better with earphones? Probably it's better now. Okay. So you could see now that permeability for potassium is higher than for sodium. It means that main ion for our membrane rest potential, it will be potassium because permeability of cell membrane for potassium higher. And it means that potassium will be easier to move outside of our cell. Potassium will move outside. Inside, something should create for us negative charged parts. Inside, our negativity created by proteins. Look, protein, negative charged, and other anions. This is big molecule, this large molecule, not able to move through the cell membrane. They could not move because permeability for them is absent. They trying to move outside, but they are not able to do that. That's why what has happened, potassium moves outside, and then more potassium moves outside, then more negative charged particles we have inside. You will have question, every exam, Krok exam have question like that. What will be happened if will be potassium more moves? What will be happen with MRP? Let's think with you and let's answer this question. So my first question, one second. My first question, what will be happened if potassium channels will be more open? What will be happen with MRP? If potassium channels will be more open, will be potassium more moves outside, and inside will be more negative charge. if will be more negative charge, it means that level of MRP will be grow up. For example, from minus 60, more negative, it will be minus 70 or minus 80. It means that MRP will be grow up. If opposite story, if we will say that permeability for sodium will be up, it means that sodium will be moves inside of the cell, and as a result, we will have with you inside more plus. This case, you will say that MRP will be moves down, will be smaller. It's mean from minus 60, it could be like minus, for example, 50. Going to be more positive. And third one factor, what is important for MRP creation, this is work of the pump. Sodium potassium pump is all time working inside of our excitable structures, and here we should say with you, what should we say, that this is special pump, electrogenic pump. Why? Because we could see that we say with you before that pump is working opposite to gradient of concentration. If according to gradient sodium moves inside, opposite it will be moves to outside because of pump, and potassium moves inside because of pump with using of ATP energy. But pay attention that this pump working for move out 3 sodium ions and move in 2 potassium ions. What has happened as result? It's happened that 3 positive, 3 plus moves out, and 2 plus only turn back to the cell. It means that this pump create more electronegativity in the cell. Let's look to the question from a sample of the question from the Krok. ATP synthesis is totally blocked in a cell. How will the value of membrane rest potential change? And here is important moment. If energy is absent, if pump is blocked, it means that this case our MRP will disappear. It will disappear. Why? Because our pump all time working for turning back our concentration to their stable state. All time we should have with you sodium high concentration outside and potassium high concentration inside. If pump will be not working, it means that sodium will move inside till the equilibrium level and potassium moves outside till the equilibrium level. And as a result, MRP will be disappear. So your answer for this question, that if pump will be stop working, it means that MRP will be disappear. Remind you next question. The meaning of depolarization and hyperpolarization. Depolarization. Then more positive substances moves through the cell membrane, it will induce reaction depolarization. It means that inside will be more positive. You see, more positive, was minus 70 and moves to minus 50 or minus 30, minus 40, doesn't matter. More positive. And other question when we are talking about hyperpolarization. Hyperpolarization, it means the difference between outside or external and internal surface increased. Hyperpolarization, more negative. For example, from minus 70, it moves to minus 80. Mechanism of depolarization. Inside should be moves more positive ions. Mechanism of hyperpolarization, inside should be stay more negative ions. How we can do that? By two ways, or go outside plus and inside will be stay more negative charged, or we can put inside negative charged, like increase permeability membrane for chlorine, negative charged ion. And we have with you example of the Krok question about this topic. 1 second. The penetration of the irritable cell membrane for potassium ions has been increased during the experiment. Increase. What change of membrane electric status can occur? The right answer is hyperpolarization. Why? I remind you again, because potassium we have plus charged and it stays inside of cell. Then more plus charged will move outside, then more negative charged proteins and anions will be stay inside. That's why then more potassium moves out, then more negative will be inside. And this name as hyperpolarization. Second question. Due to activation of ion channels of external membrane of excitable cells, its rest potential has significantly increased. What channels were activated? Same question from opposite side. If we have with you that rest potential, membrane rest potential is increased, it means that inside more negative created. If negative charge created inside, it means that we should open potassium channels. Third example of the question. The action of electric current on the excitable cell caused depolarization of its membrane. Movement of what ion through the membrane caused depolarization. Depolarization, it means that inside more positive. More positive, it means that inside should be moved positive charged particles. Positive charged particles, we are talking about sodium, of course, because look options. Calcium, we are not talking about normal excitable structure. Potassium, inside, chlorine minus, and hydrocarbonate, it's also negative charged. So we have only one option, this is sodium. And also other option for sodium question, ion channels have been blocked in the excitative cells. It doesn't change significantly the quiet potential MRP, but the cell has lost its capacity to the generation action potential AP. What channels have been blocked? This is next step what we are talking. We are moved to the question of excitation and creation of action potential. When we are talking about action potential, we are and excitability of the cells, this case we are talking about sodium channels, And let's look. Action potential it is a fast, high amplitude change of membrane potential, which provides an information transmission on distance in nervous and muscular cells. A high amplitude change we have, for example, from -90 inside of cell, it's changed to +30, +40, and after back to the -90 again. This is named as action potential. Action potential create of few phases. First phase, this depolarization. Second phase, this repolarization. And important moment when we are talking about depolarization, we should say that for reach and induce action potential, we should reach the threshold level. There's some special level for different structure. It could be different level. But after this level, we will have with you depolarization and action potential. If our irritant will be not enough for reaching this threshold level, action potential will be not arise. That's why next important name, this is threshold level. And let's look for mechanism generation of action potential. First, we have with you a membrane rest potential. In this case, for our example, it will be -70. We using some stimuli. Our level of stimulus should be enough for reach threshold level. In our case, in our example, threshold level equal to -55, -55 millivolts. It means that our stimulus should be reached from -70 to 55. How many millivolts it should be? 10, 15, 20. How much should be? Guys, no, from -70 till -55, how many millivolts? 15, right? 15 millivolts. It means that our stimulus should be not less than 15 millivolts. And when we will reach threshold level, we will have this U reaction of depolarization. This time, from this moment, we will have opening of all sodium channels. Sodium moves inside of our cell and bring positive charged ions. It will be happen till the equilibrium of the sodium outside and inside of the cell nearly +40 millivolts. It will be top of our action potential phase. After, on the level +40, +35, +40, all our sodium channels will be closed. And from this moment, we will start phase of repolarization. Due to repolarization or phase of repolarization, we have due to moves out potassium ions. Potassium moves outside from the cell and as a result take out positive charged particles from the cell. As a result, we have this phase of repolarization, and from plus 40, it's turning to the minus 70 again, to the minus 70 again. Sometimes it's happened, we have post-potential hyperpolarization or post-potential depolarization. It means that after action potential could be the same time turn back to the rest level, could be happened depolarization after turn back, and can be happened hyperpolarization and after turn back to the normal state. Okay. Our sodium channels, which provide for us depolarization phase, they has three conditions. First condition, in the state of rest, they are inactivated. They are in state of rest also. This time, they have two type of gates. Look please this channel, two type of gates: activation gate and inactivation gate. In the state of rest during MRP, activation gate is closed, inactivation is open. What do we have this time? Sodium try to move inside, but it's closed for sodium. Sodium cannot move inside. That's why we have resting potential. After activation by stimuli, our activation gates making reaction for the electrical stimuli. As a result, this activation gate will be open and we have this depolarization phase. Depolarization phase, sodium moves inside. Inside we have positive charge. When we reach plus 40, this time we will have activation of inactivation gate. They are closed on the top of action potential, and sodium again not able to enter inside of our cell, and we start process of repolarization phase. During repolarization phase, this C state of sodium channels slowly turn back to the state of A, to the normal state of rest. And after when almost sodium channels turning to the state of the rest, this time we have this new possibility to have again new action potential. One more important terms what you should remember, this is absolute and relative refractory period. Absolute and relative refractory period. What should you know this time? During absolute refractory period, new action potential cannot arise. That's all. During absolute refractory period cannot arise. During relative refractory period, repeated stimuli can cause the second action potential. But amplitude of this action potential will be less than previous one. According to the scheme, you could see that absolute refractory period from the moment of the depolarization till the point 3 on the phase of repolarization. This absolute refractory period, this time all sodium channels or open or inactivated and closed. From the point marked as point number 3, minus 40 on the phase of repolarization. This time we have one by one sodium channels turning to the state of rest and started relative refractory period. It means that some of them can be again activated and can again through them move sodium. That's why if this period we will make new irritation, we will have with you action potential but less amplitude, because not all of sodium channels are renewed to the state of rest. If this time we will make new stimuli, or this time we will make new stimuli, we will not have any reaction of the tissue. New action potential will be not erased. Okay. When we are talking about electrical potential, where we can use it for practice. Of course, we are talking about work of our nervous system. We are talking about work of our muscles. Electrical potential, electrical process moves inside of all our body. All our neural system, nervous system working only because this electrical potential. All our muscles, our movements, our somatic system, our visceral system works because of electrical potential. I already not say about our heartbeats, about a pumping function of the heart, because it's also electrical process. I will tell you that this is amazing to understand that we are working like some machine, because we are working from electric. But the most interesting that we can generate this electric by ourselves. We generate this electric and we create it and we working by ourselves. For registration, for using different method, for example, we have with you EEG, electroencephalography. Electroencephalography, this is electrical impulses from the outside of cranium, what we can registrate it from our cortex and under cortex structure. This is example you could see on the picture. EMG, electromyography, also so important for understanding functional activity of skeleton muscles. And for rehabilitation, this is one of the most important methods for registration and understanding how is working muscles, where is the problem, what part of muscles not answer for the stimuli, or for rehabilitation it could be electrical stimulation muscle for repairing, for renewing its function. And one of the example from the Krok question: in the course of experiment, it is necessary to detect muscle excitation. For this purpose, the following measurement should be made. Mechanomyogram, contraction duration, contraction stage, ion concentration, and electromyogram. Of course, this case for detect muscle excitation, we should with you make electromyogram, not mechanomyogram. Mechanomyogram, it's only we will see how is contracted muscle, force of contraction. That's all. It's not interesting for us. For us, interesting to see electrical process inside of muscle. And this could be registered by electromyography. Let's move to the next question. Parameter of the constant current impulse causing the generation of action potential. Cathode direction of electric current. You have been studied this sentence. What does it mean? It means that during electrical stimulation from outside, our electrical current all time will be moved from anode to cathode, from plus to minus. You should memorize this one question. From plus to minus always moves electrical current. The value of electric current should reach the threshold value, how I told you before, should reach threshold value, which called as critical level of depolarization. Critical level of depolarization. I told you that if we reach to the threshold level, this time we will have action potential. If we not reach to this level, if we less than this level, we will have with you or cut electrotonic potential, small short depolarization, or local response or local answer. Local response or local answer, this one value of our irritant equal to 50 or 75% from the threshold level. Like here you see minus 80 it's state of rest, minus 60 threshold. This means that this case we should use with you not less than 20 millivolt, not less than 20 millivolt. If we will use 20 millivolt, this moment will be open all sodium channels, and as a result we will have with you maximum reaction. I would like to remind you that this case it's named as low, all, or none. Single cells and single fibers working according to low, all, or none. If we reach threshold level, we will have maximum reaction, maximum amplitude. If we will not reach threshold level, we will not have any action potential. We will not have any irritation. This question so much in the Krok question, that's why please, all or none, this law is working for single excitable structure. Single, it's important. And if we reach to threshold level, we will have maximum answer of the cell. If we are not reaching this level, we will not have any answer. And let's look, here you have example of the question. What kind of example? Microelectrode technique allow registering of potential following all or none law and being able to undercurrental spreading. Specify this potential. Excitatory postsynaptic potential, inhibitory postsynaptic potential, this type of potential which we will restate upside of postsynaptic membrane. We will now talking with you about synapse and postsynaptic potential upside there. Res potential, no. Receptor potential, no. And of course, we memorize with you that action potential is working according low, all, or none. Okay. The velocity of the impulse development of constant electric current, if force of irritation increases faster, the action potential develops faster too. This is important that, for example, look, we need with you 20 millivolt for induced action potential. We can give the same time 20 millivolts and we will have maximum fast answer. Or we can give these impulses like 5 plus 5 plus 5 plus 5 millivolts. This time, as usually, will be happened adaptation of tissue and we will not have with you any action potential. Also, we have with you law of duration of irritation. The longer duration of signal, then the less force signal must be. If signal will be more long period, we can use less amplitude of impulse. It's named as chronoximetry, and so often again used by neurologists for rehabilitation, for evaluate the functional state of the neuromuscular system for people. And next picture, I would like to remind you that we have with you different type of nerve fiber. In general, we can say that all nerve fibers we can divide by two types, these myelinated and unmyelinated fibers. Myelinated and unmyelinated. Myelin, this is some substance which created by special cells. These cells named as Schwann cells. Schwann cells, this is 90% of lipids. These Schwann cells turn around of our nerve fiber. And where we have this Schwann cell, we have with you myelinated fibers. Why it's important for us? Because this case where we have with you myelin, it means that there we have with you isolation. Where we have with you fat, this area cannot erase electrical current. And for myelinated fibers, we have some distance between Schwann cell. This distance named as nodes of Ranvier or Ranvier nodes. There, so high concentration of sodium channels and our irritation will be jumped around these Schwann cells on the region which named as Ranvier nodes. Because of jumping, it's like we'll be flying for long distance, and this irritation moves with high speed. When we are talking about unmyelinated fibers, this case we are talking that irritation should be moved in every spot of nerve fibers. Why you could see on the picture animal? Because I think that is the best explanation for movements of our irritation by nerve fibers. Look, when we are talking about kangaroo, of course, you can use a frog and something else, rabbit maybe. But what is important? They can jump through the big distance. That's why they move with high speed. And we will take with you other example, snail. Why snail is important? Because snail, this animal without pet, without pets. What does it mean? It means that for moves anywhere, doesn't matter. Snail should move in every point of their way. Every point of the way. Cannot move some step through the some period. It's not possible. Every point of the way, doesn't matter speed, but important, every point of the way. The same happened with electrical current, where we have unmyelinated nerve fiber. And that's why, of course, when we are talking about this type of movement, we say that by unmyelinated fibers, impulse moves so slowly. By the way, if we are talking about pathological states, sometimes it's happened with some autoimmune disease, or it's happened during age. For old people, myelin shall begin to damaging. And because of damaging myelin shell, speedness of transmission electrical signal is decreased, and that's why people have problem with transmission signal from nervous system to muscles. They have problem with locomotion, with movements, because of creation more unmyelinated fibers. Laws of conduction of nervous fibers excitation. Anatomical and physiological integrity of the fiber. It's important. If we have with you damaging, physical damaging, of course, will be not moves of excitation. What does it mean? Physiological integrity. It means that all channels, sodium channels, will be clearly worked. If some of sodium channels will be blocked, this time our irritation will be not moves. On the base of this mechanism, we have work of local anesthesia. Local anesthetic working by the next way. They are blocking sodium channels on the nerve, and that why nerve cannot conduct irritation for some period of time. And we not feeling with you, for example, pain. This is physiological integrity. Law of bilateral excitement. Normally, in the nervous neural cell, we have movements of excitation only one direction, from hillock of axon and moves to the side of synapse. One direction. Why it's happened? Also have some question in Krok exam. Why it's happened? Because their movements to opposite side limited by sodium channels. I showed you action potential and about absolute refractory period that cannot turn back excitation to right now excited area because they're absolutely refractory period. That why can move only one direction. The law of isolated conduct of excitement. I would like to remind you that inside one nerve, we have with you so many nerve fibers, and excitation would not jump in normal physiological condition. Pathological, it's possible, but in physiological condition, impulses are not able to jump between different fibers inside one nerve. You very good know that inside one nerve could be moves irritation to different area. Even we have some nerve inside which inside one nerve we can have and afferent and efferent fibers. That's why in normal condition, irritation never can jump from one area, from one fiber to other one. Exciting happen without fading, without tiredness, and also the law of functional non-specific nerve fibers. It means that this rule using by neurosurgeries, what they can do, they can take, for example, one nerve fiber or one part of nerve and moves to other area because and make transplantation. Why? Because not have specificity in this nerve fiber. We have specificity only where, from where moves signal and to which organ moves signal. I always compare this rule with some electrical cable. It doesn't matter from where we will take this cable. If we will take it for example, for laptop, will be working laptop. If I will change it and will join to my iron, I will be able to ironing. If I will be used for, I don't know, for blender, I will use my blender. So signal is non-specific, important only from where to where it moves. The velocity of conduction of the excitation depends on depolarization threshold, action potential amplitude, diameter of nerve fiber, and present or absent myelin. Then more diameter, then faster signal. If present myelin, then faster signal. Then less threshold level, then faster signal. Then biggest action potential amplitude, then faster signal. Example of question. In course of experiment, there has been an increase in the nerve conduction velocity, increased velocity. This may be caused by an increase and concentration of the following ions that are present in the solution around the cell. Action potential depends on sodium. That's why our and around of the cell we have only sodium, inside potassium. That's why our right answer will be D, sodium. Also, let's this question we had done already. One more question. An isolated muscle fiber is under examination. It was established that the threshold of stimulation force became significantly lower. What is the cause of this phenomenon? If we are talking about threshold stimulation force significantly lower, we are talking about activation of sodium channels of membrane. Sodium channels of the membrane. And key takeaways. Voltage-gated ion channels are located along the axon hillock and axon. They open in response to the membrane potential reaching the threshold value. The rising phase of the action potential because of sodium influx. The falling phase of the action potential is a result of potassium efflux. Action potential in single cells and in single fibers working according low, all, or none. And action potential has the same high of depolarization for a given cell under typical conditions. Neuron cannot fire a second action potential during the absolute refractory phase. Neuron can fire a second action potential during the relative refractory phase, but it requires a stronger stimulus than when the neuron is at rest. The strength of the stimulus is encoded by frequency of the action potential firing. Action potential travel in only one direction, how I told you before, due to presence of voltage in activated sodium channels. And the speed of propagation signal depends on presence or absence myelin, thickness myelin, and diameter of axon, plus, how I told you, plus amplitude of action potential, plus it depends on threshold level. It depends on threshold level. Classification of nerve fibers. Yes, we say that it divides on myelinated and unmyelinated, and now already according to their characteristic. Myelinated fibers, we have A and B type. Unmyelinated, it will be C type. It will be C type. What is important here? Look, please. A type divide on A alpha, beta, gamma, and delta. It depends on diameter. All of them consists of myelin. And look please on the picture on the table, you could see here, then biggest diameter, then higher speed of conduction. The biggest diameter, this is for A alpha fibers. Like these fibers we have with you for proprioception and for somatic motor neurons. For somatic motor neurons, signal from our brain to our muscles sending by A alpha fibers. A beta, this for touch and pressure. Gamma for motor of muscle spindles. Muscle spindles, this is proprioceptors inside of our skeletal muscles. Delta, it's about fast pain. It's about feeling cold and it's about touch. Type B, myelinated fibers, it's about preganglionic sympathetic nervous system, because after ganglion we have without myelin fibers type C. Type C fibers, unmyelinated fibers. We have movement by these fibers, information about slow pain, hot, mechanoreceptors, and also it will be, look, sympathetic postganglionic fibers. Also important for information. Next lecture, I will talk with you about sympathetic parasympathetic system and their structure. I will remind you, but now you could see this difference. So we have myelinated, unmyelinated, A, B, C, they has difference not only because myelin and because diameter. Then less diameter, then less speed transmission of signal. Exam question. It was established that the conduction velocity in the nerve fibers was equal 120 meter per second. Specify the fibers. 120, this is the higher speed, and we should remember that this will be A alpha neurons. And here, so easy because preganglionic, postganglionic, sympathetic, parasympathetic, you should memorize with you. And remember that the higher speed will be for motor neurons. And this case, right answer will be E, motor neuron axons. For transmission signal from the nerve to other or from neural cell to other neural cells or from neural cell to muscle, we have with you special construction, this construction named as synapse. Generally, we can say that we have two types of synapse in excitable structures. It will be chemical synapse and it will be electrical synapse. Important moment that most of information inside of our nervous system and from neural system to muscular system transmit because of chemical synapse. But in some case, we have with you work of electrical synapse. For example, in our heart, we have with you electrical synapse gap junction. In some part of central neural system, we have with you gap junction. A gap junction, this electrical synapse, an important moment that ions moves in two sides. It means that the synapse can move signal from one side and the same to another side. If we are talking about classic variant is chemical synapse, we are talking only about one direction conduction of the signal from the presynaptic membrane to the postsynaptic membrane, from presynaptic to postsynaptic. I remind you that between them we have synaptic cleft, presynaptic, postsynaptic, synaptic cleft. Inside of presynaptic membrane, we have with you vesicles with neurotransmitter. On the postsynaptic membrane, we have with you channels. These channels will be open under action of neurotransmitter on postsynaptic membrane. As a result of opening channels, we will have with you postsynaptic potential. And I will remind you that we have excitatory postsynaptic potential and inhibitory postsynaptic potential. What is the difference? It depends on process what arises on the surface of membrane. Excitatory postsynaptic potential after summation will induce for us action potential and transmission of electrical signal. Inhibitory postsynaptic potential will induce hyperpolarization, and its result is usually its cause inhibition of transmission some signal. Steps of neurotransmission, transmission of neural signal will be is the next. I remind you these steps is also important for you. So step number 1, I would like to show you a sync video. It will be better for understanding in some action for you. 1 second, wait me please. Here we will talking about transmission signal from nervous cell to the muscle cell, but it doesn't matter. Same way will be transmit from one neural cell to other neural cell.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open.
Please pay attention for this moment. When we are talking that action potential moves to the presynaptic membrane, this moment we have opening of voltage-gated calcium channels. Calcium moves inside of presynaptic membrane. Calcium contacted to the special proteins which contact with the vesicles. And after entering calcium to presynaptic membrane, we have with you movements of vesicles with neurotransmitter. It means that this time we have process of exocytosis. Only this moment. Exocytosis provoked by calcium molecules. And this important moment.
And calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis.
Here is only one example, because when we are talking about neuromuscular transmission, We are talking only about one type of neurotransmitter. It always will be acetylcholine, always. If we are talking about transmission in the nervous system, it's so many different options we have. How I told you before, it's going to be acetylcholine, dopamine, epinephrine, norepinephrine. It could be serotonin. It could be glutamate, aminobutyric acid, and so many different options. But we are talking, for example, this is transmission from nerve to the muscle.
Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber.
I remind you, when sodium moves inside, it means that plus charge moves to inside, and as a result, we have with you depolarization. Depolarization, more positive inside. When we have in many places many depolarization, as a result, we will have summation of this process and we will have a rising of action potential.
The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in two ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
Okay. I would like to show you a real synapse. This is a real motor end plate and muscle fibers under microscope. This is a consequence how is going on this process, what we say for transferring signal. I will not repeat again. You see all steps here. All events, neuromuscular transmission. Again, what we just have been talking with you. This also a nice picture, this photo. Under microscope, you could see presynaptic, postsynaptic membrane and moment of rest period and stimulation of releasing vesicles and transferring there to postsynaptic membrane. I would like to remind you that we have with you delay of signal for during transmission signal through the chemical synapse. This delaying happened during transferring signal from the presynaptic membrane to postsynaptic membrane, because there we have with you a synaptic cleft. This cleft could be from 50 till 100 nanometers, and we need time for diffusion neurotransmitter from presynaptic membrane to postsynaptic membrane. That's why we have with you delay of signal. Delaying of signal happen because of transmission in central synapses signal. blocking of potential. I will give you example. Blocker of a signal could be, for example, curare or curariform substances. What is it? Curare, this is a substance which blocked nicotinic receptors. Nicotinic acetylcholine receptors located here, upside of postsynaptic membrane. When acetylcholine release moves to the postsynaptic membrane, contacted to nicotinic acetylcholine receptors. After contact will be open sodium channels, sodium moves to inside. If we will block with you sodium transmission because of blocking nicotinic receptors, we will stop with you transmission from nerve to muscle signal. And example from Krok exam. Curariform substances introduced into a human body cause the relaxation of a skeletal muscles. By the way, using in surgery practice for muscle relaxation. What change in the neuromuscular synapse cause this phenomenon? When we are talking about curare or curariform substances, this time we're talking about cholinergic receptors blockade on the synaptic membrane, postsynaptic membrane. Also, I would like to show you another question. A patient came to the hospital complaining about quick fatigability and apparent muscle weakness. Examination revealed an autoimmune disease that caused the disorder of functional receptor condition in neuromuscular synapse. What transmitter will be blocked? If we are talking, again, you not moves inside all this question. If we are talking about transmission from nerve to muscles, we're always talking about acetylcholine and nicotinic acetylcholine receptor. That's all. This main thing what you should memorize. Other type of blockers, tetraethylammonium blocks potassium channels. Because of blocking potassium channels, potassium induced for us repolarization, falling phase. It's induced falling phase more longer. Tetrodotoxin blocks sodium channels, and because of blocked sodium channels, we cannot induce any action potential because sodium cannot move inside. Local anesthetic, how I told you before, Novocaine, cocaine, doesn't matter, bind to the sodium channels, inhibiting it, cause the reduced number of action potential and lack of pain perception. Local anesthesia also blocking sodium channels. If blocked sodium channels, action potential will be not erased. And I will not spend so much time from you. I would like to show you mechanism of muscle contraction. I will do it using also movies, and it will be last question for today. Let's remind with you about consequence, what is going on for muscle contraction. And we saw with you first part, so electrical signal moves from the nervous system to the muscle. What is going on after? Let's look.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling.
Important moment, excitation contraction coupling This transferring electrical signal from nervous system, electrical signal what arise upside of muscle, and transferring it to mechanical process contraction.
If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse,
T-tubules.
or T-tubules, are regularly spaced in foldings of the sarcolemma that branch extensively throughout the muscle.
I remind you, sarcolemma, this is a membrane for our muscle fibers. Around of T-tubules, we have with you sarcoplasmatic reticulum, special cistern for muscle fibers. This is important because there we have high collection of high concentration calcium.
Fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal
cistern and one T-tubule together named as 3A.
cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
Pay attention, that main ion which is responsible for coupling between excitatory process, electrical process to mechanical process, it will be calcium ions. What is going on? Why they are responsible? What is going on inside of our skeleton muscle? I will remind you also.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere.
I would like to remind you, sarcomere to that line, between them we have some distance for our muscle fibers. Its name is sarcomere. We have here myosin and actin fibers. Pink color for us, this is myosin fibers, and yellow color, it's actin fibers.
Shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments.
Actin, sin, thick, myosin.
The formation of
this is molecular structure of actin and myosin. Actin, two fibers, you see, two fibers of actin. Also there we have additional protein system, troponin and tropomyosin proteins. They're included in the process of activation of actin and communication between myosin and actin. But the most important signal molecules, it will be calcium. Look, it's violet color here.
A cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. tropomyosin moves away from the myosin-binding sites on actin.
So calcium bind to troponin. Troponin change conformation tropomyosin. Tropomyosin change position, and we have opening of active center of actin. That's why now myosin head can connect it to the actin,
allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2.
I want to pay your attention also that this binding happened under angle 90 degree. It's obligatory moment, 90 degree.
The power stroke. ADP is released and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position.
Now is a magic process, magic for contraction of our muscles. Let's look again. How is complicated this process is? Look, please. So when we are talking about muscle contraction, we are talking about movements acting between myosin, and these movements is our contraction, muscle contraction. When will happen relaxation, it will be moved to the normal rest position. And look please how it happens, this sliding, because myosin walking upside actin and like touching this fiber inside.
As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin-binding site on actin.
And as a result, we have this relaxation. So we still have contraction till the moment calcium binding to the troponin-tropomyosin system. When calcium moves back, why will moves back? First, because we have working pump which moves out calcium, or moves back to the storage. And second moment, calcium will not move outside because we will not have electrical stimulation. Till the moment when we are sending electrical stimulus from our central nervous system to the muscle, we'll be releasing calcium and will continue contraction. About isometric, isotonic contraction, about tetanic contraction, type of tetanic contraction, we will talk with you next lecture. There's so many questions about this type of contraction we have in the Krok questions. That's why we will remind with you this question also. For today, lesson is over.